the pharmaceutical company Lundbeck. And I'll take you through some work we have done together with the company Atom on engineering antibody functionality and developability through iterative generative AI. But first, a few words about my company, Lundbeck. Lundbeck is a pharmaceutical company headquartered in Denmark, in Copenhagen. We have a global presence. We have research sites across the world. There's about 5,000 employees globally, 1,000 of them in research and development. Lundbeck is 100% focused on CNS-related diseases. So basically, all our targets are related to diseases of the brain. So this is our commitment. Our pipeline is 90% focused into neuro-rare and neurospecialty indications. We both do antibody and small molecule modalities, and we have 9 clinical trials ongoing. 5 of these is on antibodies, and this is of course just the tip of the iceberg. We do a lot of engineering work in research, and among other things, we do of course affinity maturation. This is a classical challenge. Aim is to drive up affinity of an antibody, usually with the aim of getting higher potency. What can happen in that situation is there is also a loss of specificity. So basically, in the process, we lose a bit of specificity, and we get some weak interactions to other targets along the way, and we term that polyreactivity or non-specificity in this case. And basically, this is a non-desired developability property. We really want to avoid this. It requires secondary engineering. Basically, it's time-consuming and it's costly. And if you can avoid it, we would like to do so. The molecular basis of the polyreactivity is of course found in the chemistry of what is the substitutions that are being incorporated in the affinity maturation campaign. And basically, amino acids that are good at driving protein-protein interactions are also good or driving up the affinity. It's also good to driving interactions to other targets. So amino acids like the aromatic amino acids, tryptophan, tyrosine, also arginine, for example, are really good at driving these types of interactions to proteins, and therefore commonly associates with polyreactivity if they form patches. But basically, the underlying interactions are, of course, governed by the general protein-protein interactions. Forces like electrostatics or hydrophobic interactions are, of course, present. So one of the reasons why polyreactivity is also an undesired property is that it's correlated to with increased in vivo clearance. So basically a poor PK profile. And of course, we would like to have a good PK profile of our antibodies. We want to have low clearance. We want to be able to dose less frequent and also to allow for lower doses. In this work by Kelly and al. They show the correlation between clearance and also polyreactivity. Polyreactivity can be measured in multiple ways, both cell-based and also ELISA-based, but it can be screened for in vitro through simple techniques. So in one of our projects, we really wanted to drive up affinity. That's even more important when you have central targets. You need to cross the blood-brain barrier. That's difficult enough with the naked antibodies. We wanted to have high affinity. We wanted to have low plasma clearance. However, whenever we affinity matured, we increased the affinity to the target. I can come closer. Whenever we affinity matured, we would increase the affinity to targets, and we would also see that the clearance would increase. So that is, you can catch it here. We have the parent, and we have some of these affinity matured variants, and we can see that the clearance here is much faster. And down here we have the half-life against the antibody KD. So whenever we sort of increase the affinity down to lower values, we would actually see an increase in clearance. And basically, what we also saw is that polyreactivity on these candidates would also go up. So therefore, it was we thought that okay, if we can decouple the two, if we can drive up affinity while maintaining a low polyreactivity, we would solve this problem, and we could actually get a good candidate that would have high affinity but low clearance. So we turned to Atom's engineering platform, and basically the concept is shown here, where we have a lead at the top. We then identify substitutions that we want to introduce, that we think that are good substitutions to introduce to get high affinity. And this would sort of give us the basis for the sequence space that we are working in. We would then distribute these substitutions out across a number of variants in accordance with design of experiment to really sample the sequence space optimally. We would then go ahead and express these molecules and purify them, and we would take them through a panel of different tests. In our case, of course, we will test polyreactivity and we will test affinity, but also other critical quality parameters such as melting temperature and titer and so on to monitor all the different functional sort of dimensions that we really want to keep track of in this study. This would then the information from these assays would then go in and inform a machine learning model. And train that model to be able to generate new optimal sets of substitutions, finding the optimal variant that we could then go ahead and express, and this would hopefully be a good molecule. Then we could then also go through the cycle again and again, and then it would become an iterative model. So the concept is also shown here of how to couple these the sequence space with the functional space. So here on the left we have this matrix with a binary matrix with different substitutions that we would like to introduce, and we have distributed those over different variants. And on the right side we have the different functional assays that we would like to take these molecules through and keep track of along the way. And basically, this simplistic model shown here, but we would be able to delineate the impact of all of these substitutions on each of these parameters. So we would have a model or several models that could inform us on how did each of these substitutions impact each of these traits on the right. So what is really critical in this type of work is to have really high quality data to put into it, because we are screening quite a large sequence space in this work. From 96 variants, we would cover a sequence space of 10 to the 15th number of combinations potentially. So basically, only a very few data points cover a very large space. So it's extremely important to have very good data on each of these variants. We went ahead and measured expression titer, aggregation propensity, thermal stability, you want to keep track of course, target affinity measured by SPR. And then the polyreactivity, which was central to us to keep track of. And there's, of course, again, different ways of measuring polyreactivity. In this case, we used an extended array of antigens that we wanted to measure on, and we took time to really make sure that this assay was in peak shape in order for us to really track the performance on it. So of course, we need to start somewhere, and basically the origin of the initial substitutions can be found in various ways. We can take approaches that goes in from sequence, looking at what can be predicted maybe from software like AlphaFold. We could also look into the alignments, germline alignments, to make sure that we also introduce things that are make sense. We have structural input. If we have structural available data available, we can of course use that from crystallography or cryo-EM on the complexes. We can also do some in silico work on those to even further understand what the impacts are. For example, free energy perturbation studies. That can also be simply experimental insights from different ways, alanine scans and so on, that can help us understand, of course, how what are the impact or contributions of some of these residues. Also, modifications of liability sites, or even positions that are related to immunogenicity would also be of importance to account for in this work. We went ahead with 2 initial rounds, and we made 96 variants, 2 times 96 variants, covering 50 substitutions. So the space that we're looking at here is about 10 to the 15th. What we found was that in these 2 rounds that we didn't really push the affinity to any great extent. We did not move the polyreactivity either. So what is shown here on the right is the KD and the poly-R here of the first round, and the second round did not manage to push the KD much, and not into the target zone where we really wanted to be. What is shown in red is the standard affinity maturation that we had done previously, and where you can see we get a good affinity, but of course also the polyreactivity is a kind of in the other end of the spectrum what we really wanted. So basically we were thinking that the conclusion was that we were working within a too tight sequence space. Here we were centered around a local minimum, and we wanted to go a bit broaden out our search to find another minimum here. So we had to look a bit further for other substitutions to include, and unfortunately, I cannot really show the structure of the complex that we're looking at. But basically, we did some reanalysis of the antibody-antigen interactions and kind of picked some substitutions slightly more distal from the actual epitope-paratope interface in order to search a bit broader out in space. So there was multiple substitutions that were picked in order to really broaden the space that we searched in. So then we went into to round 3, and here on the right we have a plot of the polyreactivity against the measured affinities on these candidates. And as you can see, now we got really broad distributions with many affinities in a range, which is actually quite good compared to the lead here. But we would also see many of them would actually have a lot of polyreactivity associated. Still, we found a few hits here that were definitely on the right path. So this looks something like this in the animation as shown before. First round is already there. The second round would not push the KD too much, but didn't cause trouble. And then the third round, we would see that now we were actually really getting increased affinity, but we would also see this broad distribution on the polyreactivity. At this point, the models we had really starting to become well-informed and also really strong. So they are really good at predicting the experimental data. And here is the dissociation rate from the SPR measurements measured, predicted against measured values. And basically in red, we have the model fit. So this is how the model that is being used to train, or the data that is being used to train the model is really being fitted, and that's not surprising that correlates, of course. But the cross-validation set is more important. These are data points that are held out of the model training, about 10% of the data, and they are also really well predicted. So the model is really strong at predicting the outcome of the substitutions. Also, it shows that the epistatic effect between the substitutions are low. So the model was really good at predicting what kind of outcome would come from different substitutions. We can also look sort of across different sort of contributions on different parameters. So here we have shown polyreactivity against the dissociation rate from SPR. And when we have on the left, we have the heavy chain, and on the right, the light chain. And these plots are slightly complicated to look at. But what's important is that double negative is really good, and double positive is really bad. So basically, we're looking for substitutions that are placing themselves in this quadrant down here. And as you can see, it's not so trivial just to find substitutions that would actually do good on both parameters. So it's actually also really tricky to find just the right substitutions to really have a gain on both. We can also look across all dimensions. Here we have some substitutions plotted on all these different assays that we have taken them through. And we have SPR and polyreactivity. And as you can see, if we would just take number one, for example, that we have often quite sort of opposite responses in the different assays. So what would drive up have a good impact on the affinity from the SPR would be associated with a negative impact across the other parameters. So only a relatively few set of few substitutions would actually have a positive impact on all of them. I believe actually that number 4, for example, is looking relatively promising. But in this way, we can sort of dissect our data in various ways, and we can track the contributions of these substitutions across the different dimensions at the same time. So then we went back into our round 4. Now with these strong models to inform us, and again we have the outcome shown here on the right. We have the KD and we have the polyreactivity, and now we have also all the parents from the different rounds. So starting out with round 1 parent here as the lead, and then our round 2 and round 3. And as you can see, we are slowly inching down into this lower left corner where we want to be, low polyreactivity and low high affinity down here. So here we managed to identify 4 candidates that are really looking good on affinity and have acceptable polyreactivity profiles. And if we look across the different dimensions down here, again, we have the round 1 plotted already, and then we have the round 2 that didn't do much to the affinity. Round 3 where we saw a lot of diversity, and then we have the refocused round 4 really being informed by these models, and from this round we could actually identify hits that would have acceptable properties, and we have achieved the goal that we set out to do. So in summary, machine learning and generative AI, machine learning informed by design of experiments, is a really strong tool to actually screen a vast sequence space with very few candidates, in fact, expressed. Here in this project, we screened the sequence space, the combinatorial space of 10 to the 27th, characterizing only 400 monoclonal antibodies in the process. And of course, the data that goes into this work is really of importance, but the limited number of molecules that needs to be screened, so 400 is not that high, then we can really nurture each of the assays, which is really important, so we get the optimal data quality on each of the data points. at the end, we identified these four sequences, and these lead molecules, they are now being progressed at the pipeline at Lundbeck, working their way through there. So with that, I'll head into the acknowledgement. We have a dedicated team at Lundbeck, and here's our collaborators at ATEM. I also want to highlight this white paper published in Bioprocess online that also describes the approach that we have taken here. So go ahead and take a look if you're interested. And with that, I will say thank you and happy to take questions.